So for the past week or so, we've been looking at the different organelles that are in a cell, and each of the specialized jobs that they have, sort of like the specialized jobs that parts of a city have to make everything sort of run smoothly. Well, we're going to take a look now specifically at the nucleus and the really important molecule that's inside of the nucleus, and that's DNA. And so back in the 1950s is when we first learned what DNA actually looks like. And Jim Watson and, the other guy's name is escaping me, Crick. So Watson and Crick, anyways, they discovered the structure of DNA and what it looked like, along with the help of another woman, which we'll kind of get into in a little bit. But in this cartoon, so as you can see from the model, the structure is basically a long, twisty, rope laddery type thing, and that really is what DNA looks like. If we zoom in on it close enough. So by the end of the PowerPoint, you should be able to summarize the relationship between genes and DNA and describe how the structure of DNA fits the function of replication. So what does DNA look like? Well, first of all, it's a polymer made of monomers called nucleotides. So that's a big mouthful. A lot of new vocab words in that sentence. So let's break it down. First of all, polymer and monomer. Polymers are just long chains of repeating units called monomers. So if you take paper clips and you hook them all together in a big long chain, the whole big long chain would be a polymer and each individual paper clip would be a monomer. And so that's kind of what it shows down here. When I was younger, my great-grandma used to make paper chains with us, where you'd take a strip of paper and you'd fold it into a circle and you make a big long chain to put on the Christmas tree, and so it's sort of the same idea. Each link in the chain is the monomer, and the entire chain is called the polymer. So now we've got the first part down. DNA is a polymer made of monomers called nucleotides. So each one of these individual units, it's not like a ring in the paper chain, it's not a paper clip in the long paper clip chain. This individual unit is called a nucleotide in DNA. And nucleotides have three different parts themselves. They've got a phosphate group, a sugar, and a nitrogen base, which actually sounds a lot like ATP, strangely enough. But DNA isn't used for energy, it's used to transmit genetic information or pass on traits to the next generation. So a long chain of DNA makes up genes. And those genes are what get passed down from parents to children, which is why we all look like our parents to an extent. So there's 4 different types of nitrogen bases, and I'm just going to point out the 3 parts of a nucleotide again so you can see them in the picture. So the blue spheres, these are the phosphate groups. The sugar is this 5-ringed or 5-pointed ring in the middle. In DNA, that sugar is called deoxyribose. That's what the D stands for in DNA. And then a nitrogen base. And like you can see A, G, C, and T, there's 4 different types of nitrogen bases. And they get put into 2 different groups because of the way that they look. So purines, adenine and guanine, they're grouped together. And I bet you can tell just by looking at them. They get grouped together because they're made up of 2 carbon rings. So this one on top is 1 ring, and this one on the bottom here is the 2nd ring. I kind of have a silly way to remember purines because I think of G-A-Ga, kind of like the sound a baby makes, like gaga. And I think of just how pure babies are and innocent, so I think of babies when I think of when I'm trying to remember which bases are the purines. The other 2 bases, you'll notice, have just 1 ring. These bases, cytosine and thymine, are the pyrimidines. And I have another silly way to remember that they're pyrimidines. So if you think about what you learned about ancient Egypt and how much they didn't really worship cats, but they were a big part of their culture. So CT, you're just missing the A, like in cat. And pyrimidine kind of looks like the word pyramid. So I think of cats and pyramids like in Egypt to remember that cytosine and thymine are pyrimidines. But you can come up with your own silly way to remember things too. I think that's probably more useful than my silly way. So how is it all put together? Usually the DNA molecule is described as a double helix. Helix is just something that's twisted, and double because there's two strands that kind of face inwards towards each other. So you've got two strings of nucleotides that are bound to another that are bound to each other in the center. So the backbone, which it's funny that it's called the backbone. Backbone's usually in the center. But the backbone is really what provides its support, and that's why it's called the backbone here, even though it's on the outside. So sugar and phosphate make up the backbone, and DNA has two. Like I said, they're along the outsides. And they have really strong bonds between the sugar and the phosphate, and that's what gives it kind of its more permanent structure. On the inside, these are the nitrogen bases of different colors. So red and green and yellow and purple, they always bind to each other. And these nitrogen bases have a pretty weak bond that's in between them. It's a very weak chemical bond because we want our DNAs to stay together, but it's going to become really important that this bond is weak when we talk about how DNA can replicate or make a copy of itself. So purines can only bind to pyrimidines, and pyrimidines can only bind to purines. And the reason that is because purines have two rings, and pyrimidines have one. So if a purine is bound to a pyrimidine, that means that two rings for the purine, one ring for the pyrimidine. That's 3 rings across. And the same with pyrimidines binding to purines. 1 ring for the pyrimidine, 2 for the purines. So again, there's 3 across. So that keeps the width of DNA uniform, so it doesn't kind of bow in and bow out. And so the fact that purines and pyrimidines only bind to each other, those are called the base pairing rules. So A always binds to T, so that's why you see red and green always across from each other. And G always binds to C, and that's why you see the purple and the yellow always across from each other. So how they figured it out, so this guy named Edwin Chargaff, he kind of ground up DNA from a whole bunch of different animals and analyzed what is this DNA made of. And he made the observation that the number of A's and T's seem to always be the same, and the number of C's and G's always seem to be the same. And in different animals, there were different ratios of A's and T's versus C's and G's. But A's and T's were always the same, and C's and G's were always the same. And so he thought, well, this is awfully odd that in every sample that I pick up, this is always kind of the rule. And so he thought, well, I guess these parts must bind to each other in some fashion. Doing separate research, a woman named Rosalind Franklin, who worked in, I believe she worked in Germany, or no, she worked in England. Rosalind Franklin took pictures of DNA using X-rays. And so this is actually a picture of the DNA. And this is sort of like if you were to look down on the double helix. And so she was an expert at reading these images. And she kind of figured out that DNA was sort of this double helix. Watson and Crick were 2 guys who were working on this issue or this problem together, and they were making models of things like coat hangers and cardboard. And they had these huge models they would hang from the ceiling and try to fit together all of the pieces. But ultimately, they weren't able to figure out exactly how it fit together without Rosalind Franklin's pictures. And without specifically her interpretation of these pictures, because they looked at this just like you and I do and say, well, what the heck does that mean? So they actually used her interpretation of what these pictures meant, not really to her knowledge. So they almost stole this information. Kind of an interesting story. So the structure of DNA kind of fits its function when it comes to replicating. So you learned that the backbone of DNA, which is actually on the outside, is very strongly bonded together, and the inside is very weakly bonded together. And so that allows these weak bonds to be broken and new nucleotides to get added. So this whole process of replication happens before mitosis. So DNA has to make a copy of itself, and then once the cell divides in half, well, now each new cell has a copy of all of the DNA. So each of the strands of DNA serves as a template or sort of a model in order to make that new strand. So the process of replication is pretty simple. You start out with a molecule of DNA, and these dark blue, it's dark blue just to show you what's old or original DNA versus what's newly made DNA. So then the bonds between nitrogen bases are broken, and those are pretty weak bonds. And then these nucleotides, which are just sort of floating around in the cell, they come and they bind to the complementary nucleotides in the original DNA. So this G is going to bind to the C, and A is going to bind to T, T is going to bind to A, etc. So now we've got our two new strands of DNA, and if you compare those strands to our original strand that we started with, the dark blue, you can see that they're exactly the same. And sometimes this is called the semi-conservative model of DNA replication because, so semi meaning that it's kind of conserved. And it's kind of conserved because half of it is what you started with and half of it is new. So you have about a meter of DNA in all of your cells. And just to give you an idea of how long a meter is about 3 feet, a little bit longer. So replication happens at many points at the same time. So down here you can see where it's sort of the twisted ladder of DNA. And then it's getting separated here in the middle. And this part where it's separating, this part right here, this part right here, those are called replication forks. And so the replication forks are where DNA has separated so that replication can begin. And there's, within one strand of DNA, there are lots of replication forks because we want this process to go fast so that our cells can divide quickly. And with over 3 billion base pairs in each one of your cells, if this was happening just at one place, that would really take a long time. So the way that replication works, just in a little bit more detail, and we're not getting into as much detail as we could be, believe me. But so the first step is that DNA gets unwound. So you can see that up here in this picture, and then there's an enzyme or a protein actually that comes in. That's called DNA polymerase. And DNA polymerase is a molecule, it's an enzyme that moves along the strand of DNA and adds the nucleotides that are complementary. It also goes through and it proofreads that new DNA for mistakes, making sure that the old DNA that the new DNA is complementary to the old DNA, that you don't have any mutations or mistakes in the DNA.